Welcome to this platform. So this video is a full coverage of chapter 2, Organization of the Organisms. That is for IGCSE Biology 0610 for the exams in the year 2026, 2027, and 2028. That is for both core and extended. So if you are writing these exams, this is the lesson for you. And of course, at the end, we are going to do the questions based on the topic. All right, so organizations of the organisms. What do you need to know about this one? So let's start. All right, so under organizations of living organisms, the first part that we want to look at is the cell structure. Okay, the cell structure. So what about the cell structure? Now, in terms of a cell structure, we look at both the plant and both the plant and animal cell. Now you know that the cell is a building block of life, or the smallest unit of life. So to start with, we look at the plant and animal cell. So let us label the parts. I guess you've watched the first part, that is characteristics and classification of living organisms, because there we did also look at the cells. But anyway, so this outer part of this animal cell is called the cell membrane. So this is the animal cell this is the animal cell. Then of course this other one is the plant cell. So this outer part is the cell membrane. Then you can point at this as the mitochondrion. So this is the mitochondria or chondrion. Then of course this one is the nucleus. I'm putting them here because I want to also look at what they have in common with the plants. Then of course here these are endoplasmic reticulum, so I'll just put ER, but what is important are these parts that we have looked at, that the cell membrane, the mitochondria, the nucleus, and also I can point at these as the ribosomes. So these tiny things inside as ribosomes. Which other part can I point at? So these are the parts that we can look at. Now, on the plant cell, this is the mitochondrion as well. Then now, the inside part is the vacuole, but I'm going to point at the nucleus as well. So that is the nucleus. So this inside part is the nucleus. Then we have this also, these are Golgi apparatus. So let me just label this as the chloroplast. So we have the chloroplast. Then we have the endoplasmic reticulum. Okay, endoplasmic reticulum, of which we have the rough one and the smooth one. How do you know it's a smooth or the rough? Because of the presence of the ribosomes. So you find also tiny things freely suspended here, which are called ribosomes. Okay, ribosomes. Then which other part can we talk of? The other part that we can talk of is this large part, which is the vacuole. Now, it's very important to know the parts that are found in both plant and the animal cell. So this is the vacuole. Now, the chloroplast, the vacuole, and the chloroplast, the vacuole, and the outer part of the plant cell is known as the cell wall. So the cell wall, the chloroplast, the vacuole, these are normally, or these are just found in plant cell. So what you need to know from here are the parts, what part they have in common, the plant and the animal cell, and what they have, the difference that they have. Of course, the plant cell has more parts compared to the animal cell. And the extra part that it has, which the animal cell does not have, is the chloroplast, the vacuole, and the cell wall. As we look at the questions, these questions or these parts are very important in differentiating the two cells. Very important. I guess you've gotten something up to this point. The cell might not come as this, but very important to understand the parts that they have in common and also the parts that they have, what makes them different. Now, let us look at the functions of the parts. So, to look at the functions of the part or structure of the cells, we focus mainly on the nucleus, the mitochondria, cytoplasm, ribosomes, and cell wall, chloroplast, and vacuole. These mainly are the ones that we focus on. So to start with, we are going to look at the parts that they have in common. So one, plant cell and animal cell, they both have the nucleus. Now, what is the function of the nucleus? So the function of the nucleus, you can say it contains genetic material that controls the cell. So you can say it contains genetic materials that controls the cell. Or you can say it stores it stores DNA and control all cell activities within the cytoplasm. So any of these, the function of a nucleus is contain genetic material that controls the cell, or it stores DNA and controls all cell activity within the cytoplasm. Then the mitochondria, that is where aerobic respiration take place. So the key thing, aerobic respiration take place, or that is where energy is produced. These questions or these parts, very important that you understand their functions, because in questions you find these part. Now, it provides necessary energy for the cell functioning. So the mitochondria is where aerobic respiration takes place, and it provides necessary energy for cell functioning. What about the cytoplasm? It is a jelly-like substance where chemical reactions take place. So that is where chemical reactions take place. And this is where all other cell organelles are found. Parts of the cell such as the nucleus, such as the chloroplast, such as the mitochondrion, all of them are found in the cytoplasm. So sometimes you find the question such as where is the chloroplast found in the cell? So it is found in the cytoplasm. So the cytoplasm is just simply the space inside the cell which houses other cell parts such as the nucleus, the cytoplasm, the nucleus, the chloroplast, the mitochondrion, Golgi apparatus, and all those. So very important that you understand this thing. Then what about ribosomes? So that is where protein synthesis take place. So if the cell has ribosomes, just know that proteins are synthesized in the ribosome. So ribosomes mainly are for protein synthesis, or that is where protein synthesis take place. Then we have the cell membrane. Now, what are the functions of a cell membrane, or what is the function of the cell membrane? So the cell membrane is simply controls the entry of material in and out of the cell. So controls what goes in and out of the cell. That is the function of the cell membrane. Now, an effective way of learning as you are going through this lesson, make sure that you have a notebook where you are able to take down the notes. These are perfect notes that you can use for your studies. Anyway, we continue. Let us look at plant cells. So those that we've looked at, by the way, these that we've looked at are found in both plants and animal cells. So sometimes you find the question, which part do they have, or which parts are found in both plants and the animal cell? So these parts that are aligned here, they are found in both plant and animal cell. Now, when we say let us look at the plant cell structures, these are specifically found in the plant cell. So you're not going to find the cell wall in the animal cell, no chloroplast, no vacuole. Now, what are the functions of these plant cell structures? So the cell wall is the outer part of a cell, and it is made up of cellulose. First thing first. The function is to protect the inside cell from mechanical damage. And also you can add to that is the one that prevents the cell from bursting when it absorbs a lot of water. So the cell wall is made up of cellulose and gives support to the plant cell. Aside from that, also protect the cell from bursting when it takes a lot of water. Then what about the chloroplast? It contains green pigment, otherwise known as chlorophyll. And that is where photosynthesis take place. So photosynthesis take place in the chloroplast. Sometimes you be given the cell and now the parts are labeled, then they ask you where does photosynthesis take place, or where is glucose produced. Glucose is produced during the process of photosynthesis. So where photosynthesis take place, that is where even glucose is produced. Then the vacuole contains cell sap and normally is used for storage. Now, aside from that, the vacuole also, it controls the amount of water or the movement of water in and out of a cell depending with its concentration. If it's highly concentrated, water moves into the cell. If it's less concentrated, that water leaves the cell. That is, the concentration of a cell sap determines the movement of water. So basically, these are the cell functions of cell structures. So you need to know those that they have in common, both a plant and animal cell, their functions, and only those that are found in the plant cell. It is going to save you marks. So very important. Now, let us look at the bacterial cell. So for the bacterial cell, it looks like this. I think it's something that we've looked at in the first chapter. If you've not watched that video, it is a variable, and also it has some questions at the end that you answer. So the bacterial cell has the cell wall outside, but not made of chloroplast. It has the cell membrane. Okay, so it has the cell membrane. Very important you understand the part. Then it has the DNA strand. Okay, it has a DNA strand. It does not have a nucleus. It does not have the nucleus, but it has the DNA strand. Then, of course, you're going to find some structures in the form of circle like this, and these are called plasmid. And because of this, it is used in genetic modification. So which structure of the bacterial cell are used in genetic modification, or which organism is used in genetic modification? In this case, we are looking at bacterial cell, and the structure, to be specific, the plasmid. I guess there's no problem up to this point. Maybe just an extra, these extension are called flagella. So we have the flagella at the end right there. All right, now let us look at organization. Okay, cell organization. Now, a cell is a basic unit of life. Now, cell can divide, make more cells, and more cells or group of cells form what you call a tissue. Then group of tissue form what you call organ. From organ, it forms the system. The system forms the whole organism. So that is how organization is. So we are saying new cells are produced by division of existing cells. How are new cells produced? By the cell division of already existing cells. And this process is called mitosis, which produces identical cells. So a single cell divide to make two identical cells, which are further divide to form more cells. So as you can see here, the process of mitosis, we have one cell here. It is preparing itself for division. Finally, it divides into two. These two cells are identical to the parent cell. Now, these two cells can further divide and, of course, form about four cells. These can divide and form. So that is how the multiplication gets to move. So cells that are produced by this process are identical, which means what they contain is identical. In other words, they are genetically identical. Now, if we are to follow it, it all starts with the smallest part, which is the cell. From a cell, it forms a tissue. So here what you are able to see is the tissue. So we have an animal cell here. Then we have the muscle cells here, which are, or which is a tissue because it's made up of group of muscle cells. Then, of course, from a lot of tissue forms an organ, and we have a liver here as an organ. So this is an organ. Then we have the system, might be the digestive system that is there, but this is a system. Then now we have an entire organism. So that is how cell organization, or that is how it moves from the cell to the tissue to the organ, system, and organism finally. So basically that is how it is. Multiple choice questions, you find which one lower and of course higher, whatever the question is, but I guess we are going to look at those questions. Specialized cells, let us look at the specialized cells. Now, when you talk of specialized cells, we know that where we are coming from, a cell is made or cells are made from already existing cells. Now, when these cells are made, when they are still small or when they're still young, they are same in terms of what they contain, shape, and also structure. But as they tend to grow, they attain specific features, undergoes changes in their cytoplasm, and that is what we call cell specialization. Simply, they undergo these changes to be suitable for their new role or the functions. So cell specialization is just a process by which the cell undergo changes in their cytoplasm or chemical component of the cytoplasm, attain some features just to be suitable for their functions. So examples of specialized cells that we are going to look at here, and what is important is to understand these specialized cells. One, the ciliated cell. So you're going to look at the ciliated cell, the root hair cell, the palisade mesophyll cells. Then we look at the neurons, red blood cells, sperm cells, and also the egg cells, otherwise sperm and egg cell known as gametes, which are just sex cells. So these are specialized cells that we are going to focus on, and very important, these do come in an exam. All right, so this is the list of specialized cells. So the first one, as you can see, this is the ciliated cells. Then we have here, we have root hair cells, root hair cell. Then these that are here, these are called palisade mesophyll cells. Then, of course, this is the red blood cell. Then this is the sperm cell. Then this is the egg cell. Then this is the neuron. So these are specialized cells that we focus on. Now, what is the function of the ciliated cell? So the ciliated cell simply removes dust and it removes mucus. Let me say it removes mucus and dust containing what? Containing germs. It removes mucus and dust containing germs from the respiratory tract. So take note of that as you are going through this part. So remove mucus and dust containing germs. That is from the respiratory tract. But these are also found in the oviduct where they move the ovum toward the uterus, and that is just move the ovum or the egg toward the uterus. One function. So they are found in the respiratory tract or in the oviduct. Then what about the root hair cell? So root hair cell. So now before we go to the root hair cell, sometimes you might be asked of the adaptations. Adaptation, we are just looking at the parts that make these to perform their function. That is sweeping out dust. So it has what we call the cilia on top there, and these cilia are able to move. So sometimes you find which part of which cells or specialized cells contain parts that move. Okay, so this cilia is able to move or beat just to sweep out dust containing or mucus containing dust and germs that is in the respiratory tract. In the ovum, they're able to move they're able to beat or to move so that they push the ovum from the ovaries. Then root hair cell. As you can see, root hair cell, normally the function is simply absorption of water and mineral salts from the ground, absorption of water and mineral salts from the ground. Now, what adaptation do they have? So they have this elongated outgrowth. Okay, this is not a tail, but elongated outgrowth to create a large surface area for the absorption of what? Absorption of water. So take note of that as you go through as well. So the main function of the root hair cell is simply absorption of water and mineral salts from the ground. Okay, water is absorbed by osmosis, mineral salt by active transport. We are going to look at that under transport across the membrane. So the specialized cells have the features which is elongated outgrowth that enables it to create a large surface area for the absorption of water and mineral salt. Also, they are numerous in number to create a large surface area for absorption of water. Okay, let us look at the palisade mesophyll. Now, these contain what you call chlorophyll, and that is where photosynthesis takes place. So they contain chlorophyll that or which absorb light energy used to split water molecule in the process of photosynthesis. So in other words, that is where photosynthesis take place. Sometimes you find a structure like the ovis, then they are labeled, let's say part A, part B, part C, then they ask you where does most photosynthesis take place. So it occurs in these cells. Now you need to know that there is a layer of cells before this, otherwise known as upper epidermis, then on top there's a cuticle. So don't mistaken the palisade cell or these that are found in the mesophyll and also the epidermal cell. In the epidermal cell, there's no photosynthesis that takes place. Instead, photosynthesis takes place in the palisade mesophyll cells. Then when you talk of a neuron, the neuron is just conduction of nerve impulse. These are the ones that makes us sensitive. So sensitivity is carried out by this one. So they conduct nerve impulse. In the case, let's say you touch on the hot object, you are able to withdraw immediately because the impulse is sent and via neurons. So neurons just for conduction of nerve impulse. Okay. So neurons, what are their adaptations? They have these spikes which are called dendrites. So these just create room for more impulse conduction. Then they have myelin sheath and also the nerve of Lanvia in between that just boosts the transmission of impulse. At the end, they have what you call neural transmitters. I think that is what you are going to look at in detail when you start looking at nervous system. That part is varied or is very important when it comes to the questions in paper 4. That is, paper 4. So you're going to look at that in detail as we look at that is nervous system. So this, we just look at the basics. Then let us look at the red blood cells. So the main function of red blood cell is just transportation of oxygen to all parts of the body. What are the adaptation? It does not have the nucleus to create room for absorption of oxygen. Also dome shaped, I mean not dome shaped, but biconcave shaped. As you can see, it is hollowed in the middle there. So biconcave or disc-like shape just to create room for absorption of oxygen. So that is the adaptation that it has. The function is transportation of oxygen and transportation of oxygen to all parts of the body. All right. Then of course, sperm cell and egg cell. These are just for reproduction. So make sure that you understand these specialized cells, 6 of them. These are very important. Make sure that you know them. All right, so the last part of this, we look at the size of a specimen and we look at magnification in this case. So they are tiny objects or tiny things that we cannot see, or those that we cannot see, that we can see rather. In other words, magnification is simply how many times is the object or the original or the actual image is magnified or enlarged compared to the image, okay, or how big the image compared to the actual object. That is what we call magnification. And normally magnification, we put x in front. So if after calculating you find 10, you say x 10. This shows how many times the actual object has been magnified. So that is what you call magnification. So magnification is calculated by using the formula magnification is equals to actual or image size over actual size. Pay attention. Magnification, image size divide actual size. Now, another thing that you need to understand, this is very important. You need to make sure that the units of the image size and the actual size, they are in millimeter. Now, if you look at the units that are used mostly, you find millimeter and micrometer. Now, this is one thing you should know. Every time you are dividing in terms of magnification, you need to make sure that the units are of the same. That is millimeter. No micrometer, but millimeter. But afterward, the answer can be recorded in whatever, sometimes magnification anyway does not have units. So in other words, here the emphasis is that every time you are dividing, make sure that everything is in millimeter. Now, let us look at the simple conversion. So if you are moving from millimeter to micrometer, if you find something like this symbol represent micro. So this represent micrometer. This represent micrometer. So the conversion is that when you are moving from millimeter to micrometer, millimeter is bigger than micrometer. So millimeter is bigger than micrometer. So when you are moving from millimeter to micrometer, you multiply by 1,000. In other words, you multiply the unit in millimeter by 1,000. Then if you are going back, you divide by 1,000. So if you are given millimeter, then you want to go back to, you are given in micrometer rather, you want to find in millimeter, you divide by 1,000. Why am I saying 100? By 1,000. Okay. So let us look at examples. And the examples that you are going to look at, we are going to do them in terms of the past papers. And of course, I can say that there are quite a number of questions that we are going to find on this particular part. All right, let us now answer the questions. So as you can see, IGCSE Biology 0610, Chapter 2, Organizations of Organisms. Exam questions from 2021 to 2025. Now, focusing on extended. All right, let us look at the first question. So here, these questions were just extracted from the past papers. Like you've seen here, that is 0610, 2022, 2025. That is variant paper 2, variant 2, May, June 2025. The first question here, which was question 4 in that particular year, what is a leaf? So this is an organization. A leaf has a lot of cells inside, palisade mesophyll cells, spongy mesophyll cells. So there are a lot of cells, and therefore a leaf in terms of organization, the level is organ. So the answer here is B. A student measured the length of a structure using a microscope. The magnification used was, so here I'll put this data here. Magnification, I'll write mg. Then it was given as 250. So that was magnification. The image size of a structure was, so image size, let me just write is. Image size was 5.0 millimeter. So it's just the same as 5 millimeter. What was the actual length of the structure? So look at this. Magnification, for lack of space, I'll write just mg. Then here, image on top, size, then divide actual size. Now, let me put magnification, which is 250. So I'm going to write the number here, 250 is equals to image size. Image size was 5.00 millimeter, which is the same as 5.00 millimeter, yeah? Allow me to put x where there's actual size for easy working out. So here, crossing multiply, we are going to have 250 x is equals to 5, which is just the same as 5. 5.00 is just the same as 5. So millimeter, then divide 250 to find x, divide 250 to find x. So x is going to be here for x, if you divide 5 divide 250, it's going to give us 0.02 millimeter. Is it what we are getting? 0.02. Yeah, 5 divide that is 250, giving us 0.02. Yeah. Now this is in millimeter. Now, if you look at the results or answers that are recorded here, we have this that is in millimeter, which is 1.25. So there's no way you're going to get 1.25 when you have 0.02. So this and this are totally wrong. Let us go for this. They are recorded in micrometer. Now, if you are to convert this in micrometer, remember that conversion from millimeter to micrometer. From millimeter to micrometer, you simply multiply by 1000. Okay, so here we are going to multiply this by 1000. So it's going to give us the answer as 0. No, it's going to give us, we are going to move 1, 2, 3. So it's going to give us simply a 20. Now, these are written in scientific form. So 20 in scientific form is just simply 1. So 2.0 multiplied by 10 to the power 1 micrometer. So the answer therefore here is C. So that is how we can work out this one. So very important. Then of course, let us look at B. This other question that is in the year 2025 as well. That is variant 2, February, March. Okay. Variant 2, this is biology 0610. That is paper 2, variant 2, the session February, March. That is 2025. SL is 65 micrometer across. So this symbol in front is micrometer across. What is the measurement in millimeter? So here we are just converting. Now we are moving, so the conversion we said from millimeter to micrometer. Going that side, you multiply by 1000. But when you are coming back here, you are at millimeter, you are coming to, you are at micrometer, you are coming to millimeter, you divide by 1000. So here we are given in micrometer, so we are here. Now we need to go back. The answers are recorded in millimeter, which means we need to convert in millimeter. So we just simply divide this by 1000. And what are we going to have if we divide 65 divide 1000? So divide 1000, and this is going to give us 0.065. So 0.065, which is the C. All right, let us look at question 6. A tick is a type of arachnids. The length of adult tick is 2.3 millimeter. What is the length of the tick in micrometer? So we are moving from millimeter to micrometer. We simply multiply by 1000. So we are going to have this multiply 1000, just 2300. That is the micrometer. So the answer therefore is D. I guess we are moving together and you are able to get a thing. Question 6. A specimen is viewed under microscope with magnification, that is times 10. The specimen image measures 30 millimeter. So image 30 millimeter. Then they are saying what is the actual size. So magnification is equals to image 30 millimeter. Then what about the actual size? Let us put X. So where there's magnification, I'm going to put 10 is equals to 30 millimeter divide, that is X. So 10X is equals to 30 millimeter. So divide 10, divide 10. So X is equals to 3 millimeter. Now, the answers are recorded in micrometer. Remember, when we are moving from millimeter to micrometer, multiplied by 1000. So 3 multiplied by 3 multiplied by 1000 is just 3000 micrometer. All right, what is the formula for calculating magnification? So actual, that is image over actual size. So image size over actual size. Question 4, that is this other question, please. So pay attention, these are coming from different years. So we are into 2024. So this other question, that the one that we've just answered comes from that is paper 2, variant 3, October, November 2024. This one is variant 1. Paper 2, variant 1, March, that is May, June 2024. So a student draws a red blood cell. The diameter of the blood cell drawn is, so diameter of red blood cell drawn is this. The actual diameter of the red blood cell is that. What is magnification? So magnification is equals to image divide actual. So what is the image size here? Is 20 millimeter. Then divide 0.08 millimeter. Okay, they are all in millimeter. So if they are not in millimeter, one was in micrometer, we need to convert them into the same unit, which is millimeter. So in this case, it's going to be 20 divide 0.008, and this should be able to give us 2,500. Magnification, you put x in front. That's why there's x in front. So the answer is C, this one. I guess there's no one who is behind. This one comes from, this is paper 2, variant 2, May, June 2024. The diagram shows an epidermal cell from an onion plant. The distance between X and Y on the diagram is, so this is the image size. The actual length of the actual length between X and Y is 150 micrometer. What is the magnification? So magnification is equals to image divide actual. Now, look at this. The image is 60 millimeter, but of course, the actual, we cannot use it the way it is because it's 150 micrometer. So we need to convert it by dividing with 1,000. So what is 150 divide 1,000? So 150 divide 1,000. And the answer is going to give us 0.2. So divide 0.15. So 60 divide 0.15 equal to 400. So the answer here is 400. So make sure that you calculate, or you also calculate. So the answer is 400. This one comes from paper 2, that is variant 3, May, June 2024. The image shows a pollen grain from a grass plant. The image has the length of, so image size, the actual length is 500 micrometer. Magnification. Magnification is equals to image divide actual size. Very important to take note of the unit. So 60 millimeter, it's okay. Divide. Now, the actual length is micrometer. So divide this one by 1,000, and it's going to give us 0.5 millimeter. What is 60 divide 0.5? So that's the same as 60 multiplied 2. So 60 divide 0.5. It's going to give us 120. So this is going to give us times 120 as magnification. So the answer is C. Which formula shows the correct way of calculating the actual length of an object? So this one comes from paper 2, variant 2, February, March, that is 2024. So they want us to find the formula for actual size. So magnification, you know that, is equals to image divide actual. So just make actual the subject, of which is just the swap. So it's just the same as actual is equals to image divide magnification. So actual is equals to image divide magnification, and the answer is this one. Question 5, which is coming from 2023. So we've entered 2023. So this is paper 2, variant 1, October, November 2023. The question, the diagram below shows the flatworm. The flatworm has been magnified by, so magnification is 100. What is the actual length? Now, when they ask you the actual length, magnification has three things. It has image, size, and actual. So now, if you have, or you are finding actual, magnification surely is given 100. Then, what is the image size? That is 50, as you can see from here, millimeter, here, this one. Then divide x. Cross multiply to be 100x is equals to 500. I guess I've not mistaken. 500, is it 500 or 50? It's 50, not 500. Divide 100, divide 100. So you're going to find x is equals to 0.5. Now, 0.5 is in millimeter. So you need to convert it in micrometer. Okay, so multiply that by 1,000. It's going to give us, I guess it's going to give us 500. And the answer is D. Don't forget to convert. Don't forget to pay attention to the units. Micro, millimeter. Let us look at this one, paper 2, variant 2, that is October-November 2023. The length of a bacterium in the diagram is 50 millimeter. The magnification is 5000. What is the actual length? So magnification is equals to image over actual. So in this case, the image size we are told is 50 millimeter. Let me just put it down here. Magnification is given as 5000 equal to the actual length is, the image length is 50 millimeter. Actual is what we are looking for. So you're going to have 5000 x is equals to 50 50 rather, not 50, 500, 50 millimeter. Divide 5000, divide 5000. Here x is equals to 5000, 50 divide 5000, 0.05, I guess. Is it 0.05? Let me just divide 50 divide 5000, not 0.5 but 0.1. Sorry for that. So 0.1. Now, look at the answers. They are recorded in micrometer. So from millimeter to micrometer, I simply multiply this one by 1000. And you are going just to move the decimal 3 times. 1, that is 1, 2, 3. So it's going to give us 10. So here, is it magnification? No, not magnification. But the actual length is going to be 10 micrometer. So the answer that we are looking for is this one. This one is coming from, that is question 4 here that you are able to see, is coming from paper 2, variant 3, October-November, that is 2023. The diagram shows the structure of parasite cell from a leaf. Which structure contain chlorophyll? You see what I was saying? Which structure contain chlorophyll? So this is the one that contain chlorophyll. These are chloroplasts, and they are found in the cytoplasm. So these structures are called chloroplasts and contain chlorophyll. The dark one is the nucleus. Question 5, the same year. A diagram of white blood cell has a diameter of, the magnification of it is, that is 8000. What is the actual length? So magnification is 8000 is equals to 76. That is the image size. Then x, that is the actual. So cross multiply. We are going to have 8000 x is equals to 76. In other words, we are dividing 76 divide 8000. By now, I'm sure we know, or you know what we are doing. So divide 76, that is 76, divide by 8000. And this is giving us, 0.9 or 0. Or let me just put it 9.5 multiplied by 10 to the power negative 3, which is just the same as 0.09512, 0.095. So we can say 1, 2, 3. Okay, 0.095. I guess that is it. Okay, so that is the same as the one on top there. Now, the answer is recorded in micrometer. What we found is just something that is in millimeter. So multiply this one by 1000 is going to be 1, 2, 3. So it's just going to be 9.5 micrometer. So the answer is 9.5, which is this one. All right, let us look at this one. The photograph shows the plant cell. That is, or this question is coming from, that is paper 2, variant 1, May, June 2023. The photograph shows the plant cell. The length of line XY is 90 millimeter. The actual length is 30 micrometer. What is magnification? So magnification is equals to image size divide actual size. What is the image size? We are told is 90 millimeter. Now pay attention. The actual size is 30 micrometer. So we cannot use it. We divide by 1,000. So 30 divide 1,000. 30 divide 1,000 is 0.03. So down here 0.03 millimeter. Then divide. What are you going to find? So when you divide 90 divide 0.03, you should be able to find 3,000. So magnification is 3,000. So in this case, where is it? So right here. This one is coming from paper 2, that is variant 2, May, June 2023. In the diagram, the palisade cell, in the diagram of the palisade cell, the distance between X and Y is 20 millimeter. The actual length or the actual width of X and Y is 40 micrometer. What is the magnification? So magnification is equals to image size. You've seen how question comes, then actual size. So we put 20. So in other words, they're just going to play around with units. 40 micrometer, we cannot use it. We need to convert it in millimeter. So divide 40, divide 1,000. So 40 divide 1,000, and this is giving us 0.04. So here we are going to put 0.04 millimeter. And what are you finding? 20. So 20 divide 0.04 millimeter, giving me 500. So magnification is 500. So which is the B. All right, let us look at this one. So this one is paper 2, variant 3, May, June 2023. The diagram or the diameter of an egg cell from a frog. The diameter of an egg cell from a frog is this. The diameter of an egg cell from a human is that. Which value shows approximate how many times larger an egg cell from a frog cell than an egg? So what value shows approximately how many times larger an egg cell from a frog cell or from a frog is than an egg cell from a human? Okay, so we are finding magnification still. So magnification is equals to image, or in this case we are going to use the size of the diameter of the egg from the frog, which is 2.5 millimeter. Then divide the diameter of an egg from human, but we need to convert it into millimeter. So 120 divide 1,000, which is equals to 0. So this is 0.12 millimeter. Then divide now 2.5. So 2.5 divide 0.12, and this is giving me 20.8, which is 21 if we are to approximate. So the answer is B. Let us look at this question that is coming from that is paper 2, variant 2, February, March, that is 2023. The actual diameter of the cell is this micrometer, so which means 7.0 micrometer. What is the magnification of the cell? What is the magnification of this cell? So which means this is the image size. So magnification is equals to, what is the image size? 49 millimeter. Then divide 7 micrometer, we need to divide 1,000. So the only thing that I've seen here is just the conversion. Okay, don't forget to convert into millimeter when you're finding magnification. So this is giving me 0.007. 1, 2, 3. 0.007, yeah. So 49, that is 49. 49, I'm dividing 49, divide 0.007, okay, and this is going to give us 7,000. So 7,000, is magnification, and the answer is C. I hope by now every one of us has gotten everything. Then this one is coming from 20, that is paper 2, variant 1, October, November 2022. The diagram shows the cross-section of part of a leaf. What type of cell carries out the most photosynthesis? This is the question that I was talking about. So the answer here is A, that is the palisade cell. The gut cell and the sponge cell also carry out some photosynthesis, but of course that is not their major functions. So the one that has major function in terms of the main function as photosynthesis, carrying out photosynthesis, is A. This one is coming from paper 2, that is variant 2, October, November 2022. The cells listed have specialized structure that allow them to carry out their functions. So the cells listed have specialized structures that allow them to carry out their functions. Do not forget about those 6 specialized cells: root hair cell, the palisade mesophyll. We talk of the egg cell, the sperm cell, the neurons, the root hair cell. Don't forget about those. So here we have the ciliated cell, the nerve cell, the root cortex cell, then we have the sperm cell. Now, which cell have the structure that can move? You remember that question? So it is the ciliated cell and the sperm cell. So it is 1 and 4. So the answer is B. This one is coming from paper 2, that is paper 2, variant 3, October, November 2022. And the first question here is, root hair cell are found on both plants. Root hair cell are found on plant roots. Which feature is present in root hair cell but not in the sperm cell? I think this question answered it in the first part, that is characteristics of living organisms. But anyway, so the one that is found on or in a root hair but not in the sperm cell. So chloroplast is not found in the root hair. Cytoplasm is found, but also this one is found in sperm cell. Cell membrane is found, but also in sperm cell. The only thing that is not found in the sperm cell but found in the root hair cell is the cell wall. What is an example of a tissue? So a tissue is made up of a group of cells, and in this case, it's not a chloroplast. Chloroplasts are cells. Neurons, cells. Pancreas, not cell. But here, the answer is palisade mesophyll cell, or palisade mesophyll. All right, so this one is taken from paper 2, that is variant 2, February, March, that is 2022. The diagram shows a mitochondrion. The scale bar shows the actual length or actual size of the mitochondrion. So this, they are saying this is the actual. The bar shows the actual. The diameter is 70 millimeter long. The diagram shows the mitochondrion. Scale bar shows the actual size of the mitochondrion. The diameter or the diagram is 70 millimeter long. What is magnification? So magnification here is equals to image size divide actual size. Now look at this. What is the actual size? Actual size, they are told, is 1 micrometer. Then, so magnification is going to be, image size is 70 millimeter. This actual is 1 micrometer, so divide by 1000, and we are going to have 0.001. Divide here, what are we going to find? Should be able to give us 1000. Or 70 divide 0.001. This is giving us 70,000. So magnification is 70,000. Two types of cells, one animal and one plant cell, were examined using a light microscope. Which row shows the correct combination of structures that they would have observed in the cells? So animal and one plant cell. Animal cell, plant cell. So chloroplast, there's no chloroplast in animal cell. Cytoplasm is there, nucleus is there. There's no cell wall. there's no chloroplast, so which means it will be cytoplasm, nucleus, chloroplast in the plant cell and cell membrane. So the answer is B. We look at this one, that is paper 2, variant 1, October-November 2021. The length of the mitochondria in the photograph is 15 millimeter. The actual length of this is 3 micrometer. What is magnification? So magnification here is going to be the image size is 15, that is the photograph image, divide, the actual is 3 micrometer, divide by 1000. So 3 divide 1000, still going to give us 0.003. So divide now 15, divide 0.003, and we are going to have 5000. So this is millimeter. We are going to have 5000 magnification. So the answer here is C. The diagram below shows some cells. I guess you're able to remember these cells. Where are these cells found? Not alimentary canal, not blood, not plant roots, but in the bronchus, which is the respiratory tract. I guess you remember I said they are found in the respiratory tract or oviduct. So the answer here is C. This is paper 2, that is variant 2, October-November 2021. The diagram shows some tissues in part of the root. Arrows represent the movement of water in and out of, in and across the plant root tissues. How many different tissues are there? Now, a tissue is made up of cells. So these cells are root hair cell, one tissue. These cells in the middle here are cells, group of cells, another tissue. These cells that are moving like this, another tissue. Then we have where the water now is moving, which is the xylem, another tissue. So here the answer is B. So 4 tissues. Question 4 of the same paper, length of a mitochondrion in a photograph is 15 millimeter. Then the actual length is 3 micrometer. I think this is something that we are just from answering here, which is right here. Okay, so this is the same question was just repeated. So in this case, the answer is C, which is 5,000. Let us look at this one. That is paper 2, variant 2, October-November 2021. The diagram shows some cells. Where are these cells found? I think this is something that we've answered already. So it's found in the bronchus. What about variant 3, paper 2 of October-November 2021? The length of a mitochondrion in the photograph is 15. Actual length is this. So this one also we've answered it already. That is 5,000 as magnification. This one is paper 2, variant 3, October-November 2021. Okay, this paper, I think some of the questions were repeated. If not me who was repeating the same questions, but I guess you understand that we've answered this one already. This one is paper 2, variant 1, May, June 2021. The diagram shows some of the blood vessels, other structures in the human body. The blood vessels shown are all parts of the same dash, cells, organ system, tissue. So these that are shown, as you can see, we have the heart, this from the blood connecting to the heart. So this is called circulatory system. Okay, so are of the same what? System. Question 6, the photograph shows a plant cell nucleus measuring 2 millimeter across. If the magnification of it is 500, what is the actual size of the nucleus? So magnification is equals to image size divide actual size. So magnification is the one, or we are finding the actual size. So magnification is 500 is equals to, the image size was 2 millimeter. The actual, we are going to put x, so we are going to have 500. 500x is equals to 2 millimeter. Divide by 500, divide 500, and we are going to have x is equals to 2 divide 500. So 2 divide 500 is giving me 0.004, 0.004. That is what I'm getting. Now this is in millimeter. Was it recorded in millimeter? Yes, which is this one. All right. This is paper 2, variant 2, February, March 2021. The diagram shows cells as seen under the microscope. What will be present at X? So here at X, what will be present? Is it one cell membrane, two cell membrane, one cell wall, or what? So this question is yours, and I guess, and I hope, I think this is the last question anyway. This is the last part rather that we are going to look at. So make sure that you answer this question, including this one. So this part, I leave it up to you to answer. So the diagram shows the root hair cell. How is this cell modified for the absorption of water? So these questions, you are going to answer them, okay? These you are going to answer them. They are your questions. So be able to put your answer in the comment section. And of course, please, if you've enjoyed up to this point, it has been a long journey. Do not forget to subscribe to this YouTube channel. And also, you can also register for membership on this platform, where you can receive the video specifically for you, and also other nice deals. So do not forget to register for membership. All right, let us look at this part, that is paper 2, variant 2, February, March 2021. This is just the continuation of this, I guess. As you can see, it's question 6. So there's also a question on magnification. Now, I've done also a lot of questions on magnification. I would like you to try out this question. So make sure that you try out this question as well. And I believe in you. You can't fail to answer this question. All right, so we've come to the end of the presentation. So please do not forget to subscribe to this YouTube channel. Do not forget to invite others. Otherwise, this platform, you are going to revise topics from the Cambridge IGCSE in biology, chemistry, and physics. So stick around, and you are going to benefit greatly as you prepare for your exams. Do not forget to subscribe, and see you in the next one.